I am going to give this last lecture, and it's not in the usual tradition of this course, in that usually the course coordinator gives the last lecture, but Jeannette has graciously offered to let me do it because Biology 336 in this term is the last course that I will teach in a regular way at UBC, because I'm about to retire. So this gives me my chance to sort of tell you a little bit about some of my perspectives on biology and how I have been as a biologist. And the who part actually matters. And I think that that's one of the points today. We are humans doing this work. We are not just automatons. So, okay. You know what these are. You know, in the course, we've every so often had a what is this, but you already know what these things are. You've seen pictures of these. These are jumping spiders, the group that I work on. And this is a course about evolutionary biology. And just like all biologies, evolutionary biology is diverse in its approaches. We use mathematical models, we use lab experiments, we use field work, we use phylogenetic patterns. But we're also diverse as people in our inclinations. We like details, or we like the big picture, or both. We like the outdoors or the indoors, or both. We focus on ideas or organisms, or both. We do pure applied research, or both. We work alone or in teams, or both. Or we seek fame or understanding, or both. And so this is about the who, in a sense, and I want to tell you a little bit about me. And it's not to say that I'm the center of the universe, but it's to put a focus on different approaches. Every one of you, as to how you approach science, are going to bring something different to it. And so this is a little bit, some of this lecture is about what I bring to it and how I respond to my work. Actually, I just remembered, I'm going to take this off. And I'm going to take it off for two reasons. One is this is my last lecture. I'll take it off. The second reason is that I just got COVID about a month ago, and supposedly, and I'm negative now, so don't worry about that side of it, but it's still affecting me a little bit. I'm probably a bit low energy, but it also means that I'm not going to catch it, at least not right now. So, okay, so this is going to start in a place called Divide Creek, and this goes back in my history quite a ways. At this point, I was 12 years old. And I am the one in the red sweater. My brother is the one in the green sweater. That's my father, or our father, and a first cousin. And we are near the border between Alberta and BC. And this is not the photograph that I would have cared about. It represents that day, but what happened that day otherwise was something that was indelibly etched in my memory. But we don't have a photograph of that place, that other place on that day. And what it was, we went to this place called Divide Creek. But I managed to get back there a few years ago, and so I finally took a picture. And so here is me at Divide Creek, considerably older. And so what's so special about Divide Creek? It's on the Continental Divide. That's why it's called Divide Creek. But what that means is, the particular way it's on the Continental Divide is that instead of being tributaries that fuse, it's a single creek that splits. That is right on the Continental Divide, that point of splitting. And so you'll see the arrow in the bottom there. The blue arrow represents the flow of the stream. It comes along, it hits that little pile of rocks, and it splits into one branch that goes to the Pacific and another branch that goes to Hudson Bay. And I remember as a 12-year-old, and this was probably the moment that if I thought about it, I would have known that I was somehow doomed to be an academic. I thought, wow, just imagine two little bits of water floating along, a millimeter apart. They hit that pile of rocks, and one of the little bits of water happens to bounce to the left and goes to the Pacific, and another little bit of water happens to bounce to the right and go to Hudson Bay. From such close proximity and conditions, those two bits of water end up having such different destinies. And that was like, how strange that little bits of chance can have such a big effect. And that's what I thought when I was 12 years old, and it was like, this is mind-blowing. And I think every one of us can think of Divide Creek moments in our lives at which either we make a choice or some little thing happens that changes what we would have done otherwise. And so for me, one of the big Divide Creek moments was the following year when I was 13. I was on the shores of Lake Ontario with my brother, and we were looking around for things on the shores, and a mat of grass floated by. Presumably some household had mown its lawn and just, they didn't compost in those days. They just threw the mat of grass onto the lake, And sitting on that mat of grass was a jumping spider. And she was a fairly big one, big enough to notice as she was floating by, and she looked up at me. And that was so strange. I looked down at her. I don't think her eyes twinkled, but my eyes twinkled, partly because it was so strange to see this little creature reacting to me and looking up at me. And so I just got really into jumping spiders. And I think one of the reasons that they're so fascinating to me is because they react to the world in a way that feels familiar to a visually oriented creature like myself. I sort of see what they're looking at, and I have some idea as to what's going on in their head. Probably not really, but I think I do. Whereas most other little creatures, they go bumbling around and I don't quite get what they're up to. So I started looking not only for other jumping spiders of this species, but also other types of jumping spiders, because I think I have this deep fascination with diversity. I also loved being in the field. I loved going out into the wild and looking for things. So here I am in Cypress Hills, Alberta, in 1977. It turns out that I found on that day an undescribed species that I haven't yet got around to describing, but I'm hoping to do this year. It led me to many places, this quest for different jumping spiders. To Mexico many times, to Ecuador quite a few times, to Gabon once, only once to Africa, to New Guinea, an amazing place, to Borneo. And so I got to see so many different environments. And oh, the one I should have added is Siberia. Oh my God, southern Siberia is amazing. But not only was I just loving being out in the field, but I loved to illustrate them. To me, this illustration brought out my artistic side, but it was also a way to celebrate the creatures. So these are the two oldest drawings that I can find, that I know, from when I was 14 years old. And looking at them, I can actually recognize the species, which isn't too bad. But my drawings were more than just documentation. So for instance, as I continued with this, and this was from when I was in high school, I guess, the drawings that I did more than simply capture form. There was a little bit of gratuitous beauty involved, right? I just wanted to show people how amazing these creatures were. It really mattered to me to celebrate it. But of course, I did the more technical, straightforward documenting illustrations along the way. This is a long time ago. I'm getting a little less patient as I go. So the drawings aren't quite as intense. This is 2020. But I'm still doing it, right? And in fact, this is my favorite thing to do in science, apart from being in the field, is to do little drawings of spider genitalia. And so I actually have this video here that you can look up. You can just search how I draw jumping spiders on YouTube and put my name in there. There's the URL, but it's hard to write down. So from this background, from this set of interests, drawing jumping spiders, getting to know diversity, being out in the field, I became a spider taxonomist, going out and finding new species and describing them. I also became a systematist, trying to understand how they fit together with one another in an evolutionary way, working out the relationships. I also became, along the way, a theoretician, thinking about how we sort this all out. How do we do it? What's the theory of phylogenetic methods? And then, of course, I had to work to try to implement some of these methods and thoughts, and so I became a programmer building analytical tools. So on the outside, what you see when you look at my scientific record is basically the sum of my publications, right? That's what a scientist appears to be. And publications tend to follow standard formats, and so most scientists seem more or less alike, I think, probably. But on the inside, a scientist is the sum of the motivations, joys, and stumbles that propel them to do the work. And everyone is so different in this, or at least the potential for a lot of diversity, that scientists, in fact, are much more diverse than their papers might lead you to believe. And for me, I think probably the most distinctive thing is that as I've done this science, I also see and think about and appreciate the beauty of these organisms. I can't help but do it. It sort of infuses itself into my work. And so for me, these are not just study organisms. They're basically found works of art. They're things of beauty. And not only is each beautiful alone on its own, each of these is the naughty bit of a jumping spider male that is of different species within the same genus. So these are all the different species in one genus, and I drew them and arrayed them like this. And I remembered when I first built this arrangement of all of these, it was like, whoa, not only is each like a little abstract sculpture, but together there seems to be movement there. There's something special about patterns and diversity. So they're beautiful. The diversity itself has a certain dynamic beauty to it. So I walk into forests not only for the science, I have to admit, but also for the beauty. And I think most scientists don't talk about this very much. Maybe we are starting to talk about it more now that we have social media, but I think almost all of us feel this way. Okay, the unexpected. This is the tree of life. Well, not really. It's just a simulated thing. I didn't want to try to draw the real thing and everything. But imagine that this is the tree of life. Jumping spiders are here. You are here. And we can now think about the connection between us and the jumping spiders. And we can think about that set of ancestors going back along our lineage into the past, the set of ancestors along the jumping spider's lineage going into the past. And we can, in our mind's eye, find the point where they meet, the most recent common ancestor of spiders and humans. And we can also even imagine what it might have looked like. And unfortunately, we don't know. It would have been some sort of little bilaterally symmetrical wormy creature in the sea. I'll just draw it like this, okay? There you go. Just imagine that this is one of the species, a member of the species, that is the most recent common ancestor of humans and jumping spiders. And you can then take your mind's eye back to that ancestral species, or let's just think of it as a little population. Of course, it would have been likely bigger than this, but let's just show it like this. And what I'm showing here are the generations of descent of this little wormy species that is our ancestor in common with the spiders. And I'm showing the blue lines being the parent-offspring relationship. So each of these worms has two parents, two lines coming from it, going into the past. And so this is the passage, the blue lines are the passage of genetic information, generation by generation. And we can then, in our mind's eye, fast forward and think about the point at which there starts to be a divergence. And this is not, it would not have been this clean, right? So you can imagine that there might have been something, this reproductive community, this genetic community that was the ancestral species, got sundered in two for some reason. It might have been that some subset got blown off by a current to an island, another shallow area. It could have been that there was sedimentation quickly that divided the rocky habitat in two. We don't know what it would have been. It wouldn't have been this clean. It wouldn't have been this fast. It would have been messy. But in some way or another, there must have been something that took this reproductive community and split it into two. And if we look at any such diagram like this, with a set of ancestral generations becoming split into two, there must be some point in that generation, in that diagram, at which a mother worm finds that her daughter worms are on either side of this split. That one of her daughters went to the left and one of her daughters went to the right. And of course, you're seeing Divide Creek right here. So now let's look forward in time to the consequences. In this case, it's not the Pacific and Hudson's Bay that they go to. Of course, it's hundreds of millions of years later. One side diversifies into many creatures, including us. The other side diversifies into many creatures, including the jumping spiders. And there you go. From those two offspring of that one mother, what different fates they ended up having, or the fates of their genes. And so this raises the issue, is evolution at all predictable? Just think about this. Those split things, they had such different fates as to who they became in the end. And remember that ancestor didn't have legs, may not have had eyes. So basically all sorts of stuff had to evolve. And so there's this old quote by Stephen Jay Gould, a paleontologist and evolutionary biologist who wrote a lot of thoughtful pieces on evolutionary biology. And he said at one point, imagine winding back the tape of life. This is back when there were tapes. Imagine winding back the tape of life to the early days of the Burgess Shale, around the time of the Cambrian explosion, and just say, okay, now we're going to start time again. Would it come out the same way? Would we get humans coming out? Would we get whatever coming out? How predictable would it have been back then as to how evolution turned out? And his point was, it's not very predictable at all. We wouldn't have expected that humans, or something like humans, or he said it, something like human intelligence, would end up evolving in that way. And so he's basically saying evolution is ridiculously unpredictable. And so he asked this as a thought experiment, and in a way we already know the answer of sorts. We know the answer because the Gould's tape of life has been played more than once. At each of these splits is basically saying, okay, we're now going to set two things up. We're going to set two sister lineages up, and each one of them is going to be an independent replicate. Do they come out the same? And I know it's not exactly what he would have imagined because, of course, there's the potential for interaction between them. He would have had multiple replicate Earths if he was going to do this for real. Like if it were conceivable to go back in time and set up replicate Earths at the exact point so they wouldn't interact. But I think we all look at this and we say, oh yeah, for sure. If there had been a third thing, it wouldn't have come out either as a jumping spider or a human. And in fact, we do know a third thing, right? A third thing would be some jellyfish or something like that, right? So it came out very differently the second time. And so again, is there any predictability? Is evolution chance and chaos? So the bottom line here is that evolution is deeply contingent. It's full of chance. But as scientists, we don't really think it's that simple. We believe in regularity. We believe in pattern. And so the question is, should we? Are we just misleading ourselves, seeing patterns that don't exist? And the thing is, when I look at these spider palps, I don't see chaos. There's integrity there. There's something, there's a pattern there that somehow makes sense. Okay, I got to keep telling you about jumping spiders, right? That's sort of the theme of my lectures. I've shown you this group before, Hibernatus, diverse North American. Males have cool ornaments on them, colors and so forth. But I'm going to take you to one particular place and a particular group of Hibernatus. These are the southern Arizona sky islands. So Arizona, you're in your mind, you can imagine the Sonoran Desert. But the mountain ranges that are in the south get up to an elevation such that the forests at the top resemble the forests of British Columbia. So fir trees and so forth. And along the way, in the middle range, there's like oak trees and so forth. But because there's desert in between, these things are like little islands of the sky. And they have acted more or less like a Galapagos Island set for some creatures. And the one that I've studied is this one, Hibernatus pugillus. You can find them on their little rocks. They're fairly dull looking from the top. When you look at the males, you realize that each of the mountain ranges, so this is southern Arizona there, Tucson, I don't have my little pointer, Tucson would be sort of right in the middle, more or less, or a little towards the top. And you go to each of these mountain ranges, to the oak woodland, and you realize they look different in each one. In one of the mountain ranges, they've got little eye streaks that are half there. In another mountain range, the eye streak is full. In some of the mountain ranges, the faces are silver, and so forth. And so there's been this differentiation, even though the spatial scale is only a few dozens of kilometers. Turns out that they also have different courtship behaviors. So here's one of the courtship behaviors of one of the forms. This is one of the silver-faced forms, and he does this real quick jiggling with his palps, and then he flicks with his first legs. And then there's this other one that has a really distinctive courtship display because he takes those little palps and he waves them in circles. But he waves them in circles alternately, right and then left, and right and then left. And so there's this interesting contrast between these two forms that one of them has a more or less symmetrical palp wave, just sort of like this, and the other one has this very carefully slow asymmetrical one that's contralaterally alternating, first right and then left. And it turns out that that contrast you see again and again on the phylogeny of Hibernatus. So here is Hibernatus tarsalis from Bishop, California. This is the one that is conducting Beethoven's violin concerto. And he's just jiggling the palps up like this, right? Whereas this one from the near Yuma, the Colorado River, does this, right? So again, contralaterally alternating. And here's another species that's just on its own, and it has this really exaggerated left-right motion of the front legs. So what does the phylogeny tell us about the evolution of these, what at first seems strange, contralaterally alternating asymmetrical motions? Because there are arguments that sexually selected traits should be symmetrical, and these don't seem to be. In fact, they're emphatically not symmetrical. Well, it turns out that contralaterally alternating asymmetrical motions have evolved frequently in this genus. Of the palps, of the first legs, of the third legs, it's a pretty frequent thing to happen. So it's happened many times independently. So this is a challenge suddenly. Why would this be? Especially if some people studying sexual selection think that symmetry should be favored. Well, one possible explanation for why they evolved these motions in the courtship is just that it's genetically easy. They've already got the template for walking in their nervous system. They can, left, right, left, right, left, right. They can just take that template and co-opt it for making palps move, for instance. But it could be, though, that it's a good genes indicator, right? Like, it's like, look, you're seeing this palp move, but what, of course, that means is that my nervous system is really great at doing left-right motions, so your babies will be really good at walking and running, right? Of course, those two are not necessarily mutually exclusive. In fact, they could be closely tied. The very fact that it's available for co-option means that it's relevant, in a sense. There's a third possibility, though, that it's got nothing to do with any of those. It's just that it keeps the female entranced, right? It's like, oh my God. And there are reasons to believe that if you keep her confused in just the right way, you'll start to communicate little bits that might make her say yes. So we don't have any clue right now as to why. And in fact, this is the point at which I would hand this off, a study about this, to somebody who's much better at looking at the details of how spider brains are wired and so forth, right? But the key is that the phylogeny, the fact that it's convergent, the fact that it's so seriously convergent, that's what gave us the indication. That's what is our sort of prospecting tool. And it can tell us, look, this is something we're studying. Okay, you'll notice that I'm starting to build towards the title of this talk, which is The Music of Evolution. Again, Hibernatus. I can't shake Hibernatus. It's just really tough to shake Hibernatus. It turns out that not only do they have really cool courtship and really bright ornaments and everything, they're variable in chromosomes. And one of the things that's the most distinctive about their variability in chromosomes is whether or not they have a Y chromosome. So we think of, mammals typically have XY chromosomes. Spiders typically have no Y chromosome. Typically, they have 2 X's in the male and no Y, and the females have 4 X's. So it's basically 2 plus 0 makes 2, 2 plus 2 makes 4, and that's the distinction between males and females. It's the number of X chromosomes. So that's what you see at the right. That's a male doing spermatogenesis such that the 2 X chromosomes go to the same side. On the left, though, is one where there's a Y chromosome. And it turns out that the Y chromosome, and there's a lot of good reason to believe this, evolved by chromosome fusions. Chromosome fusions with some of the autosomes, the non-sex chromosomes, right? And this is the little diagram showing how it happens, but the basic idea is that by having a fusion between an X and an autosome, the homologous pairing of that autosome becomes automatically a Y because it goes to the opposite pole, right? So you don't need to know the details and everything, but it turns out that this likewise is scattered phylogenetically. And we've got really good phylogenetic data to build the phylogeny of Hibernatus, so we're quite sure of this. And you'll notice that the dark blue, which are the ones with 3 X's and a Y, and the green with 2 X's and a Y, are scattered around the tree. And if we try to figure out by either parsimony or likelihood what the ancestors had, there are between 8 and 15 origins of a Y chromosome in this group. And this is really dense. It's unusually dense for this sort of evolutionary change in chromosomes. So why are Ys evolving so often in this group? And here you'll start to notice the repetition. We talked about it in lecture, but it's a theme here as well. So why are there so many fusions that make a Y? And before we think about that, let's just step back and say, okay, what's the total set of fusions that seem to have happened in this group, where we can identify them? And unfortunately, we don't have like chromosome mapping, and that might reveal more fusions and more messing around, but just looking at numbers and so forth, there might not be any other fusions involved. It's a little tough to tell. There's a little set of the phylogeny where it could be that there's some autosome fusions. Maybe there's XX fusions, but whatever they are, they're not generating a Y. So there might be up to 3 other chromosome fusions implied by the phylogeny. So this is the sort of the summary. There are between 8 and 15 autosome X fusions between an autosome and a sex chromosome. There are between 0 and 1 that are autosome, and between 0 and 2 that are XX fusions, right? So there's uncertainty there. But however you cut it, those numbers are really strange. And the reason they're strange is that if you were to choose 2 chromosomes out of the chromosome complement to fuse randomly, you'd expect an autosome, because there's 13 autosomes and only 2 X chromosomes. There's 26 autosomes total, but 13 pairs and 2 X chromosomes. If you were just going to pick 2 randomly, almost always it would be an autosome fusion, right? So this is not a random set of fusions. And in fact, the chance you'd get an X, that many X, and so what I've done here, the orange circles there, are the most conservative scenario of counts, like as few Y chromosome evolutions as possible, as many autosome as possible according to the data. And if you were to choose that, the probability that you'd get such a bias towards X autosome fusions is really tiny if you were to do this randomly. So it seems that there's a really strong bias towards X autosome fusions. So why? Why would there be such a strong bias towards that sort of fusion either occurring or surviving? It's hard to know, right? But resulting in something that characterizes the species. And it turns out that there's actually an argument. There's a proposal in the literature as to why this can happen. And I'm going to tell you the story as to why it might happen. We don't have the really good data, although for some other systems where they've looked at this sort of question, they do have the good data to actually say, oh yeah, this seems to be really what's going on. But I'm going to tell you a possibility and tell you why it's so credible in this group. So this is an idea that goes back at least to the 1950s. And the idea is that there can be sexual conflict, i.e. a conflict between male interest and female interest, in a sense, regarding the alleles at some genetic loci. And it's pretty simple to think about. Like, you can imagine that there are some traits that in a sexually dimorphic species would really be advantageous in a male, but if they showed up in the female, it wouldn't be a great thing, like a red face, right? A red face, oh, that's a good thing for a male Hibernatus to have, but the females don't want red faces. They don't want to be more visible to birds and so forth, right? So if you imagine what would be advantageous for a male and a female, they could be different alleles at a locus, for instance. And if there is a gene that does that, that controls something like face color, you can imagine that these different alleles would have different values to males and females. So this is how it connects with the chromosomes. If there is sexual conflict within a locus, so here I'm showing 2 homologous chromosomes, right? And then at one locus, it's going to show up on both chromosomes. At one locus, I've shown you the little blue allele. Let's say that makes red faces. And then the pink allele, excuse the stereotype, makes white faces, i.e. better for female. If there then, once you have a system like that, if you then have a fusion between the X chromosome and one of the autosomes, that can actually be advantageous because it resolves the conflict. It lets males and females do their thing. And the reason it does that is because depending upon recombination and crossing over points and so forth, the male can have its red face, but that red face, because it's on the Y chromosome, never shows up in the female. And the female can have its white face, right? Obviously, this depends upon a lot of genetic details, but this is the basic theory, right? So maybe these Y chromosome evolutions are points of resolving conflict between males and females about sexually dimorphic traits. And the thing about Hibernatus is that males and females differ in so many traits. And each of those traits, when it evolved, when the mutation happened, say, for a red face, was an opportunity for conflict. And so I'm going to sort of show you how many traits are involved in this sexual dimorphism, right? How many differences there are between males and females. And I'm going to show it with this species. Hibernatus cicatus is very closely related to something that lives in the Okanagan, for instance. And so I could tell the same story about that. It's just that I have better diagrams and pictures and stuff for this species. So if you look at the details of the males, these are the traits that the males have that the females don't have that are presented to the female during courtship. And there are so many. We can basically imagine that we can sort of divide them into almost 20 traits. So there's the white spots on the fangs. There's the red face. There's this little line above the eyes. On the first leg, there's this funny fringe of spoon-shaped hairs. There's a couple of broad spatulate spines. There's a spot. There's some special hairs. There's a bump with orange on it. There's et cetera, right? It's like there's so many morphological differences, and probably these are all under different genetic control, or at least there's a lot of genes involved, and every one of them coming in might have been an opportunity for conflict. The behavior that the males has is extremely complicated, right? They're shaking legs in different ways. They're bouncing the palps there. It's a long sequence of many, many steps, many special behaviors that the male does during courtship. And so just to show you how complicated this courtship is, when we tried to publish this because nobody had published a good description of this courtship before. We tried to publish it in 2012. How are we going to describe this, right? We thought momentarily about using dance notation, because there's a special notation as to how you do dances. But of course, it wasn't designed for 8 legs and 2 palps and stuff like that. So we invented our own notation, and it's a little bit like a seismograph where these are the XY coordinates of different major bits and where they're moving. So it includes the first legs, the palps, and the third legs. And these are the first 2 stages of courtship behavior. First 3 stages, right? So they do something and then they change their position, and then they do something else and they change their position. These are the first 3 stages. So that's the first 3 stages. This is the 4th stage of courtship. This is the 5th stage of courtship. This is the 6th, the 7th, the 8th, and the 9th. It's amazing. It's like a Beethoven symphony, right? It's just stunning how many bits there are in the sequence of motions that the male makes. And it turns out that it's not just the motions. The male's singing as he's doing this, right? So the green stuff that I've highlighted, this is the acoustics. Or it's actually seismic because it's probably substrate-borne for the female, not airborne. And so this is what you hear. It's ridiculous how complicated this courtship is. And of course, every one of these things, bits of behavior, bits of morphology, is a trait that when its genes came in, could have been disadvantageous to females were it expressed in females, right? And we don't know whether some of these would automatically only show up in females or whether some would leak through. But there are indications that some of these courtship ornaments have little ghost traces in the females. So each new trait that evolves in courtship could be better for males and worse for females, or vice versa. And with so many traits in courtship, it seems likely that there were many opportunities for sexual conflict between loci. And it may have been then that the resolution of that was through, for at least a few of these, was through these Y chromosome generating fusions. So this is a hypothesis, right? So we have sexual selection generating more and more distinctive courtship traits, many of them. With at least a few of them, there's interlocus conflict between the sexes, leading to chromosome, which was resolved by chromosome fusions. Now we've got those chromosome fusions fixed in place, holding the alleles apart. So this might be why there are so many Y chromosome fusions in Hibernatus. And it turns out that the species group that has the most fusions has also got the most complicated courtship, turns out. But of course, life is not that simple. This is seems ridiculously complicated. How are we going to get a beat on this? It's actually more complicated than that even still. Turns out that these X autosome fusions are correlated with shifts in where crossing over points are occurring on the chromosomes, the chiasma localization. Whenever there's a shift of, whenever there is a fusion, there's a shift in where chiasmata are. So, and it's been hypothesized already that that's sort of an important thing. So this may be what we've got. We've got not only chromosome fusions coming from the conflict, but chiasma localization happening at the same time. And the key here is we have different traits evolving with consequences on other traits. So it's like there can be these little sequences of things that might have happened. So we have chromosomes evolving, repeatedly making fusions, consistently biased towards the X and correlated with chiasma localization. We have courtship having many traits being evolved by sexual selection, repeated evolution of asymmetrical motions. And I talked about asymmetrical motions, but there are other things that repeatedly evolve. And these things are possibly linked, right? And so you'll notice I'm talking about repetition. There's something about predictability here. There's repetition, there's pattern, right? So does this contradict this sense of unpredictability that I gave you when I talked about the divide creak of a dividing lineage? And the answer is actually no. And of course, this is basically something that's well known in science, that even though the fate of any particular lineage could be unpredictable, an ensemble of lineages together can show a predictable pattern. It's in a sense a predictable distribution. And one of the places in science where this is so well known is actually in the gas laws. You can basically not predict the motion of individual gas molecules, so the individual bits are unpredictable. But en masse, as an ensemble, they become completely predictable, and you got your good old PV equals nRT, right? So there are certain cases where the distributions become predictable, even if the individual bits are unpredictable. Okay, now I'm going to hit music of evolution directly on and pull this together. So I mentioned to you that when I look at this, I see this not just as individually beautiful things, but that the diversity itself is beautiful. And when I look at this, it feels like a dance, right? I look at these forms and I look at diversity, and it feels like I can feel the dynamics there. And so diversity's dance, how do we think about it? Evolutionary biologist basically asks, to what music do they dance? What tempo? What melody? What harmonies? What repetition? I could have thrown in counterpoint there too, but whatever. Also, what surprises? And so I tend to look at phylogeny, and I think many of us have this feeling, even if we don't express it quite this way, looking at phylogeny as this dynamic thing. And it's like music in the sense that there's a time sequence, there are things that happened, and each lineage, as it branches, becomes a voice in this choral work, and it sings with notes changing through time, and perhaps that sequence of notes that it sings in different traits, in chromosomes and courtship, follows somewhat predictable patterns. It's a melody. It's something that shows up again and again. So with chromosomes, perhaps the fusions and chiasmal localization follow each other, and it's repeated in different lineages. There's rhythm there. And it may be that the melody and counterpoint are sort of like continued beyond that to these other traits, that courtship behaviors and the ornaments, though evolution of those may provoke the changes. So this may be the first step in that little melody. And so we have these repeated sequences along phylogeny's branches. And so I remember when I was trying to express phylogeny to a group of people who were academics, senior academics, but not biologists. And I was trying to say to them what it felt like to me to think about phylogeny. And this was what came to me, what I could tell them. It felt like there was a symphony going on, right? There were all these voices, these sequences, this sort of predictability, and yet sparks of uniqueness, surprises happening everywhere at the same time. And I think this is one reason that many of us love to actually hold biodiversity in our mind and sort of see all the different spiders or all the different whatever it is that you study, and see it arranged on a phylogeny and get this sense of pattern, get this sense of the music. We almost feel sometimes as if we can hear this music. So I tried to do this, like I tried to explain this to these non-biologists, and then I realized, I was writing out the talk, and oh my God, I'm a programmer. Maybe I can make the music happen. Right? Maybe I can actually turn it on. So I thought, okay, well, what would it be? It would be like you're reconstructing the ancestral states, and you're making those ancestral those changes along those lineages make noises. Right? So here was what I tried to do. Okay, so let's just do the first one. Okay, I have to. Oh, please. Oh, you can. So these are the speciation events. Each little branch point, each divergence is a little tone. Right? Next is the chiasmal localization. Right? So the shifts towards more or less terminally localized. Oh, I think I have to. Okay. And you'll notice that there's lots of possibilities here because it's a continuous character. So it's not just the note of divergence or not. It's like, are you going up? Are you going down? Are you going up or down a whole lot? And it was at this point, as I started to hear this, that I was starting to think, this doesn't sound exactly like a symphony. So then there's the evolution of the Y chromosome itself, and this is using stochastic character mapping, if you know those sorts of things. And so each evolution of a Y chromosome fusion is going to make a little ping, and then the losses make a different ping. That's one that's off the screen, so you didn't see it. Okay, so now this is everything. You can imagine how hopeful I was when I hit that button, right? So now, there's a couple things wrong with this. The first thing is, why do you think, does anybody have an idea as to why it is that it just seemed to get busier as you went? Or what was the mistake I made by letting it get busier as I went? Any ideas? Any thoughts? You seem to be.
The most information for just the last bit.
That, I think, okay, so that's an interesting take on, I think, yes, there's much more information about the last bit, and there's much less information because there are all these things that are extinct, right? All these lineages that are extinct. And so I'm not representing the extinct branch points that never led to the present, right? That never led to information that we now see. I could have done that with a simulation model that sort of actually did that. So that's the first thing that's wrong. The second thing that's wrong is that I think that actually to hear the symphony, you need the voices to be different instruments so that they're not, all the sounds aren't muddled. So what I needed to have done was made it so that at that ancestral divergence, it was the woodwinds and the strings, for instance. And then the next thing was the bass versus all the smaller ones, and then the cello versus, et cetera, right? You know, and then maybe, anyways, I realized at that point that one has to sort of like really start extracting, and probably in a sense the music is not going to be the simple changes. It'll be something like the shifts in the parameters of character evolution that are behind the changes. There might be something that's a simpler model that underlies this. But I gotta say that I still believe in this. Even though it didn't sound right, you know, it didn't come out, I still believe that there's something really there about the patterns that we see when we look at phylogeny, and it's compelling. And so what I want to say to you is that even though I really talk about the beauty of the spiders, evolutionary biology is not just beautiful for the shapes of its organisms, but it's also beautiful for the shapes of its ideas. So I'm going to talk about beauty now directly. So what generates beauty? Is evolution creative? And I think the way to answer that is to think about how we are creative. And whatever you think about your intentions, I think almost anybody that's creative realizes that ideas come from who knows where. And that it's a really typical process that we sketch and then we crumple. We throw things away. We propose and we judge. There's the old saying, write drunk, edit sober. You know, you have crazy ideas and sane reflection. That the source of ideas is distinct from the judgment of those ideas, right? And that our creativity involves this, like, who knows where the ideas come from? You go on a long walk, something passes by or whatever. Or maybe, you know, you took some mood-enhancing substance or whatever it was. But then you have this point where you have to judge. But of course, this is what science is too, right? It's really not that distinct. We think of artistic creativity as being something different, but scientific creativity is in the exact same model. We hypothesize and then we test. You know, you could also equally say, hypothesize drunk, test sober. And of course, what that is mutation and selection. That's effectively what it is. Our creativity, both as scientists and artists, isn't that different from the way that natural selection has built all these amazing creatures. And I just want to take a little aside here to say, why does it matter that science is done by diverse people, right? Well, this right-hand side, that's the judgment side, you can say that it follows cold logic. And that's how we define science in terms of the testing, right? The weird thing is we define science in terms of the testing, but that definition forgets the left-hand side of the equation, right? And really, this side is about idea generation. And idea generation is not something you only do at the first part of a study. You do it throughout a study, because at every point when something goes wrong, or you're trying to decide on some subpart, you're having to think about the alternatives that might have been, right? And just as natural selection is built on a diversity of mutations and gene combinations, human creativity is built on a diversity of ideas from a diversity of people. And that doesn't follow a script. And it means that different backgrounds, different experiences, different approaches, different cultures and everything can have a substantive impact on the course of science, also values, through that, in a sense, mutation step, on that creating of ideas step, even if the test step is fairly rigid. And one of the reasons I say this is actually to talk about me, is that, like, I tell you about beauty, and it might seem I'm stepping outside of science, but not necessarily, right? Because even conversations about beauty can be important to generate the ideas that are then the raw material for the selective process. Okay, so getting back to nature, after 4 billion years of creativity, is it surprising that life would have produced beauty? Not at all. Each one of these species, at least to my eyes, has a beauty all its own. And each of these species also, as a scientist, for me, gives us data on what evolution can produce and has produced. Each species is an actor in Earth's ecosystems. Each species has evolved solutions to problems we might share. Of course, they've been trying to figure out life for the last 4 billion years, just as our lineage has. And each of those problems that they have to solve, whether it's about perception, whether it's about dealing with parasites, whatever it is, it may be that some of the solutions that their creative process of natural selection has built could be accessible to us as solutions for our own problems. And so this jumping spider, I did this. This is like one of the few times I've ever seen a drop of venom on the fang of a jumping spider. It's a little hard to see, but there's a little glint on the fang there. People are looking at spider venoms as possible sources for novel neuroactive pharmaceuticals. So species have both beauty and utility. What is their future? So sometimes when I go out into a forest or out into a desert or wherever, I find something that I've never seen before, and I think, am I the first person to see this species alive? And yeah, sure, there's a little bit of pride there, right? But it's also a feeling of being honored by this chance to be there at this moment where, at least in terms of an awareness, humans have encountered this species for the first time. But it's really hard not to also ask myself, and will I be the last? Because there are certainly places that I have been that I found new species that when you look at Google's satellite view right now, the place is gone. It's a parking lot, it's a whatever. And so I think about how all of us imagine going back in the time. Because of course, I bet how many of you have imagined like going back to see a dinosaur or trilobite or something? Come on, some of you must have, right? Yeah, of course. And so Calvin did too. He says that the reason he wants to go back is to get photographs to sell to paleontologists, because then he'll make a ton of money. But I think we really know that Calvin also just wants to see them, right? He just wants to see what they looked like, right? And so we imagine this. This is a lost world. And we yearn to see these other ways the world could have been. And I think that in 500 years, we're likely to wish that we had time machines to come back to now, right now, right? Because there is still a substantial portion of the biodiversity on this planet here. There are still pockets of rainforests, there are still good deserts, there's still good Arctic habitats, right? They're shrinking fast. And of course, they're all under threat of global climate change, but they're individually shrinking fast because of land use, right? And it's very possible that in 500 years, we will be substantially in a post-biodiversity world. I think that the chances of that are really remarkably high. Not to get too far into sort of religious imagery and so forth, but effectively the Garden of Eden is still around. But it's just about to go. So I have a confession to make, which is that I actually am a time traveler. And this photograph is particularly relevant, I think, at this point, because this was in 2011. And we were in eastern Ecuador, and a tree had fallen because of a windstorm. We could hear it fall, right? One afternoon. And we knew that if we got there the next morning, there were likely going to be jumping spiders on that tree that otherwise we'd have to go up into the canopy, which is really hard to find. So we knew there was likely a sample of biodiversity that was going to be there just long enough until the spiders figured out that they were now too close to the ground and then wandered back up into the canopy. So the next morning we went out there and, oh my God, it started raining. And it was like, but there was such a sense of urgency to me, right? I had to go through this tree to find what jumping spider species were there that I was never ever going to see again, right? And that sense of urgency is what drives my whole career. So I managed to get this grant from, since I'm from 25, 23, I managed to get this grant, take a time machine back to more or less now, right? And so, but it means that I have only a limited time, because what I want to do is I want to, just as you would do for big dinosaurs, like the non-avian dinosaurs, I want to go back and bring some stuff, right? I want to, at least I can get photographs, right? So that in 25, 23, I can show my colleagues, this is what they're like, right? It's amazing. Like, this is such an opportunity that I've got now to get that data back into the future before it's gone. Because of course, nobody was around to do it for the non-avian dinosaurs, but I can do it for the jumping spiders at least. And actually, to tell you the truth, there's a whole batch of us around that are doing this too, right? We haven't all told people that we're these time travelers, but we do it. The problem is that the time machine only works in one direction. It can only go into the past. And which means that I'm not going to see my family again. I'm stuck here, right? But I can get the specimens into the future. They're not going to be alive, but this is how I get them to the future. These are the museum collections, and I'm hoping that the specimens last 500 years, right? But just think about it. Even if we only had pickled non-avian dinosaurs, we'd be thrilled right now, right? Just imagine what you could see, and photographs. They'll get photographs too. The photographs will survive 500 years, I presume. So my mission as a time traveler is to make sure that we at least have these 500 years from now. But of course, it would be so much better to save this, right? To have these 500 years from now. The problem is that how do you convince people to take the actions that they need to take to save that, to keep it around, to make it so we're not in a post-biodiversity future? And I think that, I guess I'm old enough to be cynical enough to think that our ethics do not drive our desires. We do not have an image as humans as to what we should do, and therefore that's what we do. I think we actually do it the reverse. I think we just have whatever desires we have, and we retrofit our ethics to justify our desires. I just think that that's the way we mostly work, to be just truthful. Which means that our best hope actually is about desire. Our best hope is to fall in love with this earth and care about it, to give value to it. And the key is that it's not just we biologists. It has to be everyone. Biologists love it, but we're such a small fraction of people that we don't have much effect. And so this is why I talk about beauty. To be scientists, we must share the truths we uncover, but to be responsible, we must share the beauty we behold. So thank you for exploring evolutionary biology with us. That's it. Thank you. And thank you, my colleagues up there, everywhere. Thanks for coming. Do you have any questions before Jeannette talks about details?
These slides be posted to Canvas.
I can do that? Yes. And that way you can get the URL of the talk.
What would you do after retiring?
Oh, mostly I'll just be drawing spiders and going to the field. That's exactly it. I get to do exactly what I started doing. I get to return to being a high school student in the summer. Because that's what I would do every summer. Every summer was going out and catching spiders and drawing them. I had a little microscope. And every winter was, just doing schoolwork. And so I get to do that. And the cool thing is, with a lot of scientific disciplines, it's really hard to retire and keep up doing what you do because of the equipment and money and so forth involved. It turns out that there is this level of work that we can still do that's still really useful about discovering and describing the species that can be done on a small budget, a relatively small budget, right? And with a microscope that I can have in my house, right? So it means that I can actually do productive research in a little office in my house. And I think that's such a luxury to have a field where I can do that and still be me, right? So I actually feel that, like some people, they say, oh, you're going to retire, you're going to not know what to do. I actually feel like I'm going to be more me when I retire than I have been for the last, 40 years or whatever. Yeah.
Can we get like some spider highlights or something on Canvas, like your favorites?
Oh my God, favorites. Oh, that's a really interesting possibility. Oh, okay. I'll try to do a few, and maybe what we can do is maybe I'll try to do it on Piazza,
Okay.
and then you can then sort of encourage me to keep putting more on, like with dialogue. Yeah.
When, if you can take that time machine and go back to the beginning of your career, what would you do different?
I think that's a really tough question. In general, I'm not built to think about regrets. It's not how I tend to think about my life or anything. But of course, one has wisdom and thinks about what one might have done differently. I think that the biggest thing would have been is to have had more courage at certain early points as a professor to insist that I wanted to be who I am. Because there were moments where, this is a whole other talk about what's valued in science and how you could look at what I've just said and say, oh, that actually doesn't seem to be even science, or it doesn't seem to fit the model of science that we normally have, and so forth, right? Like, where's the hypothesis that I started with, and so forth. And so there's sort of a hegemony of a particular view of science. And I encountered that, and I actually think that it's really important that there are lots of different types of scientists, like there are lots of different approaches to science. But that canonical one that I was sort of channeled and told I had to do it this way, right? I didn't resist it enough. It turned out okay, right? And maybe there was some good there about like keeping me pushed in certain directions and so forth. But there was about like 10 years in there where I think that it would have been great if I had taken 20% of my effort and shifted it from here to there. Basically more on the dedication to the spiders. But other than that, I feel really, really lucky. I feel really lucky that I've been able to keep doing this stuff, right? Like there still has been a percentage of my time I've been able to go out and look at spiders. And there's just nothing better than that. So one of the lessons is that even though I feel like I didn't do it quite right, is finding something that you really love to do and letting yourself be you and actually sticking up for it can be really important, right? There's only, you only have one life, so you can hold off, so I'll just wait a little bit, I'll be me next year, right? But put a timer on that. I think I want to ask Sally that question. I would say for sure now, right? But I don't think we have a good enough sense of equilibrium pre-Anthropocene to answer that, like what it would have been before we showed up on the scene. But for now, oh wow, yeah, there's no question it's going to be really fast. And I don't think I think there's going to be probably tipping points at which just things suddenly collapse and it just becomes completely obvious that like you say, oh, this thing went extinct, oh, that thing went extinct. It's not going to be long before we won't be able to even talk about them. It's like, oh, another 10,000 went extinct, right? In the last 6 months or something, so.
So I guess kind of following up with that, like what do you think the future of evolutionary, like biodiversity research is going to look like?
The thing is there are so many, it's tough to know because there are certain things that I do that I don't think are sustainable on a really intense time scale. So the drawings I do are getting replaced by digital images. But my hope is that there's always going to be sort of a little bit of a handcrafted approach and an individual approach, because the thing that I get from these drawings is that I learn and understand better. So I think I really understand the structures better than somebody that just took a digital image. And yet, is there time to understand? That's sort of the weird thing. Like, does science permit understanding anymore? Or does it only want results? And but I think there's this, looking at the younger people, your generation, I think, there's a craving to get back to that authentic and understanding, right? And so that's my hope, that keeps a balance, right? That regardless of these pressures for result productivity, that there's another pressure that's coming from you that's about a dedication to understanding and an authenticity and a connection to what it is, a respect for what you're looking at. And that's the thing that I didn't talk about, right, is that when I look at these species of spiders, when I do a description of a new species, I really do feel as if I'm doing it for the spider, right? There's a really important part of what I do that's for that. I'm giving them a voice because suddenly they're on the map. It's known where they are. They can participate in decisions about conservation and so forth, right? So I'm sort of talking about attitudes and shifts of attitudes rather than actually what the science is going to look like. I think there will be really rapid methods for surveying biodiversity and so forth. And I mean, genomics is having such a huge effect, so I think that we'll be able to really understand things much more quickly. But that just may simply accelerate our ability to construct memorials before they're dead. So.
I was just curious, were any of the jumping spiders that you handled like dangerous to humans?
There are none known to be, okay? They're all poisonous, they're all venomous, in the sense that they've got a venom and it has an effect on insects, right? And it almost certainly, if it was in big enough dose, would affect us, right? But most of them can't even break skin, they're too small. I've been bitten twice. The one time was by something that looked like the one with the venom drop there. And the 2 times I've been bitten, a very big one, and it was because I goofed, like I went to reach and I sort of partially squashed it or something. It didn't just come and bite me, right? That almost never happens with spiders. And both times it felt like about half as strong as a bee sting, and it lasted about 15 minutes. And that was all. That was the effect. So I could feel that there was venom there, but that's the effect. So other than that, like, we don't know of any of the jumping spiders that are dangerous to humans. But no one's gone out and, okay, I'm going to get bitten by this one. Now I'll get bitten by that. No one's done that yet.
Wayne, I just want to say 2 things. One, it's an absolute honor to have been here to see you deliver this really, just fascinating and insightful, big picture, beautiful lecture. Number 2, I wanted to ask you, so thinking about statements like 10,000 species going extinct in 6 months, kind of sort of thinking ahead to mass biodiversity loss, and given our student population has grown up with this legacy being foisted upon them, you're at this later stage in your career. Do you want to offer some perspective on how people can get involved in trying to avoid that future, or some kind of sliver of hope? Because
Yeah.
what I notice is our student population is bombarded with these kinds of perspectives, and then left with that grief that they don't have a way of processing from leaving our science classes.
Yeah, for what it's worth, I think I lost my microphone a while back, but sorry for the Panopto people. So, I think the one thing is that things are unpredictable. Like, I made it seem as if we're doomed, right? But there are a number of things that I would have predicted when I was starting science. It didn't turn out that way at all, right? It's like, oh my God, we never believed we could figure out phylogeny that well. Like, there were people that said we shouldn't even try, it's pointless. But we're pretty much nailing phylogeny now, right? It's just amazing. And I think that there are lots of points at which what we'd expected was so wrong so often, right? Which means that I think we might be fundamentally wrong about the course of the future with the right interventions, or with the fact that, the next pandemic will reduce our population size in half, and so, whatever. but I think that there is hope just because we don't know what are the critical points, right? And what can change and how attitudes can change and so forth. So I think that there really is the possibility for something big to change. I think the biggest thing that can change is attitudes. I think that that's like if there actually was a broad will among the generations that are inheriting this mess, I think it could make a huge difference. What was the first thing you asked me about? But I'm not. I don't think I'm the one to. I think the thing is, the solution. I think like, the main thing I can say is I think there is hope, just because so many surprises we've had in the past, right? But as for solutions, I'm not the one to figure it out. Like, I'm one of from the generation that caused the worst problem, right? You know, you're the ones that are going to figure it out, and I believe it. Like, I believe that if there was a chance, it's amazing how impressive your generation is. Like, I gotta say that, and like what you know, I'm so glad I'm not on the job market now. Oh my God. You know, to compete against these young biologists and what they already know and what they grew up with knowing and what they're prepared to understand, I can't be the one that suggests solutions, because I really think that we're in a world where there's so much creativity now, and the fact that people are communicating on social media about themselves as people, about values and so forth, it's a complete shift from when I came into science, where you just didn't talk about these things. You were just, you know, it wasn't about values, it wasn't about choosing, right? But that conversation is here now, and I think that that's a really important thing. But thank you, Bridget. Okay, I think probably we should let Jeannette have her 10 minutes of whatever she needs to talk about.
This is just business stuff, so if you're a guest and you want to get up and sort of make your way out, that'd be totally fine. So I'm glad I decided not to try to do a short lecture before Wayne's class, because I think the value for you is really in being here, and I almost get speechless whenever I see this lecture, and I hope that you mark this moment in your memories, because there really are very few people who can do that kind of lecture, and you know, it really is an honor to be here for this lecture, Wayne. Every year, this year especially.